Yes, so let me start the session. So, good evening to everyone. Welcome to the day 8 session on molecular talking. So, before proceeding to today's session on molecular talking, can we recap the last session topics? So, first I can start the code. So, the first question is which is not a force field? You remember yesterday we discussed about the types of force fields and energy minimization. So here I mentioned the force fields and only one is not a force field. Which is not a force field? So the choices provided are CHOP, CVFF, Muki, Amber. Okay. So we have still 15 seconds left out. So which is not a force field. So we have around 21 participants at present joined in the group. So only three votes, only for found three answers. Yes, come fast with your answers. Just 10 seconds. Now 5 seconds. So we received only 8 responses at present. So try to increase your responses, 9 references. So the time up. So the answer is the majority of who answer like Mugi is the correct answer. You know the charm comes under classical force field. It also comes under polar force field. You remember, and consistent valence force field comes under classical force field, where it uses the Newton law of motion to determine the total force. Okay, and if you see Muki is a drug likeness filter. Amber is once again comes under classical force field as well as polar force field, polarizable force field. Okay, now we have discussed something about force fields. Can we discuss few more questions? Like what are the types of energy minimization we have discussed? Energy minimization methods. Today we'll be discussing on how to perform molecular talking using autotalk. Okay, there also we are using the energy minimization methods. So one should know because this is needed for talking, simulation, and other purposes, including molecular modeling. If you see in homology modeling, energy minimization is one of the method which is inbuilt, which is present virtually. While doing modeling, even energy minimization is happen. The purpose of doing energy minimization is to make the protein more stable during the folding. Okay, protein folds. If protein folds, then obviously can find the stability can occur. If protein unfolds, then stability will be loses. For this, we need energy minimization. Very good. Only one has answered. Very good. I mean, even I can appreciate that a biomedical student has tried the correct answers. So even the biotech students, I found a lot of students joined in this. So interact well with your answers. Very nice interaction from your side. So now let me go ahead with today's session because we have much more contents to see in molecular talking. So molecular talking today we discuss on what is molecular talking. The other is ligand protein talking, protein talking. Even we can talk any biomolecule with protein. Biomolecules like carbohydrate binding with protein, lipid binding with protein. Okay, because this carbohydrate, lipid, the DNA, RNA, so all these things are small molecules when compared to protein molecules. Okay, in all the cases, protein is the macromolecule, also called as receptor. And we have exclusive softwares. Like whatever we can use the tools for docking ligand protein can't be used for protein docking. Similarly for DNA protein, RNA protein, other biomolecule protein docking. And we discuss on theory of molecular docking and scoring methods and hands-on experience in autodock. Now, let me take you first to the molecular talking. So, what is molecular talking? It is a computational procedure to predict the conformation Yesterday also we discussed about conformation of a protein ligand complex. So what do you mean by conformation? We'll be discussing in subsequent slides, illustrated with pictures. And if you see this molecular docking, generally it consists of two components. One is search algorithm, the other is the scoring function. The molecular docking can be performed with free open source softwares as well as commercial tools. Okay, if you see Discovery Studio, Scroidinger, all these things, if you to go to the perpetual, it goes even beyond 20 lakhs. So then we can able to do even these kind of a docking using the freewares. Then what is the difference between the freeware and the commercial tool? The commercial tool has highly advanced search algorithm, have plenty of scoring functions. This is the reason the commercial tool is more applicable even to publish for high quality impact factor journals. Now let me take you first on the molecular docking, that is a ligand protein docking. You can able to see a ligand is docking in the particular site. Okay, this is the functional group actually. This particular functional group is binding in this site and another functional group is binding in this site. Okay, so this is what we call it as docking, and this docking we check the affinity of docking through binding affinity. Okay, that is through estimation of binding energy. So the energy whatever we calculate will be in give free energy. Now let me take you to the protein ligand docking. We have plenty of tools, nearly 200 tools are there. In 25 years before, I was using this UCSF dock, RAM dock, ICM dock. But so at present we can able to see the docking like ligand fit dock. The ligand fit is the module present in Discovery Studio. The Discovery Studio is a product of BioVia. BioVia is a company in Japan, has this kind of a tool, okay, that is Discovery Studio software, like how you find in MS Office, Word, Excel. Similarly, we can see in Discovery Studio, ligand fit docks one ligand with one receptor. If this is the module present in Discovery Studio for docking a single ligand with single receptor, for docking multiple ligands, like how we tried with Autodock PVRX, okay, with the receptor. So for this, we can use library dock, that is libdock. And if you see this score evaluation in Discovery Studio, we have one module called DS ligand score. So if you see in Autodock, the Autodock can able to generate only one score, that is binding energy. The binding energy, whatever it generates, it's in kilocalorie per mole. But whereas if you see in the ligand score module present in Discovery Studio, this can generate around 8 different scores. 8 different scores. Each and every score is a correlation of different physicochemical properties of ligand as well as the ligand receptor complex. Okay, so this is something about the modules present in docking small ligand with the protein. We also have modules to dock protein versus protein. We use some docking like CZ dock to dock protein versus protein. Okay, so like this, if you see Scroidinger, like however we discussed BioVia Discovery Studio, Scroidinger is also a commercial package. Here we have plenty of tools. For example, if you want to talk a ligand with the receptor, we use Glide tool. Got it? So like this, we also have protein docking, and even Discovery Studio has QSCR module. Similarly, Scroidinger has QSCR. So this is a collection pack. This is a package where you can see a collection of drug design methods. You can practice with almost all the drug design methods. Discovery Studio, Scroidinger. These are well-known commercial tools for drug design. Now let me take it to the protein talk. We have mostly online tools to talk protein versus protein. Okay, the scientists preferably use Class Pro, ADOX, X, etc. However, we'll be seeing the subsequent slides. Now about different talking methods. If you want to go for practicing the talk, uses of talk, some 20 years before I tried with silicon graphics interface. This is the type of OS. Now presently, if you see the talking, we are practicing either through Windows. Majorly we practice through Linux. The reason behind why we practice talking in Linux, The output of the docking files can be provided as an input for simulation, MD simulation. The output of docking is the input of simulation. So we need a kind of a single operating system which is compatible to do both. Autodoc, this is what we'll be discussing about today, and this is available in scripts.edu website. Okay, this is one of the tool available, and actually this comes under even MGL tools at present. MGL tools, MGL tools possess this Autodoc, and we have Affinity, Flex, Gold, Fred, Glide. Okay, so these are the other ligand protein docking tools. Now let me go ahead with docking. Already we have seen what is docking. We are going to see another definition. That is, a docking is identification of the low energy binding modes of a ligand with the active site of a receptor whose structure is known. For example, if I have a ligand 1 and ligand 2, and ligand 1 and ligand 2 I'm going to try to dock with receptor. Okay, whatever I'm going to do, if ligand 1 is giving minus 9.5 and ligand 2 is going to give minus 8.6, which is the best ligand? Ligand 1 or ligand 2? This is my question. My question is, I have two ligands. I'm going to bind with a single target receptor. The ligand 1 is going to give a binding affinity of minus 9.5 kilocalorie per mole, but whereas ligand 2 is minus 8.6. Then my question is, which is the best ligand? L1 or L2? Come fast with the answers. What is that? Can someone unmute an answer? Which is best? L1, L2. Or if I have L3 as 7.2, which is best? L1, L2 or L3? Come fast, participants. So this is I told you what is molecular docking according to definition. Now I'm asking you, if I try to target all the three ligands in a single receptor, I got three different energies. Which is the best one? Am I audible to everyone? Yes or no? Yes? No? What happened? Are you listening?
Sir, audible, sir.
Oh, you. Madam, what is this? No participant is interacting. Yeah, participants, why can't you? Either you can type the answer in the chat box.
So L1
You are right, madam. The answer is
minus 9.2.
yes, ma'am. So I think you got the answer from the low energy binding. So you got it. Like even other participants now understood, I believe. So whichever is negative maximum, that much is the best bind. And few participants have tried installing the Autodoc, and I know that you are facing some trouble with Autodoc or in Discovery Studio Visualizer. Definitely, even we will be discussing about that. Okay. I also, if time is there today, I ask you to share the screen, or else tomorrow I'll be asking you to share the screen. So be ready. Try to share your screen. After sharing your screen, I can clarify any kind of doubts. Okay, today or tomorrow we can try. If not today, because lot of things are to be covered. And interaction, see, for interaction you have taken this much time. Try to reduce the time in interaction. Steps in docking. The first step is the ligand preparation. The second is site characterization. The third is the scoring grid calculation. The fourth is docking. So these are the general steps in docking. It can be a commercial tool or freeware. These are the general steps we take. Now let me take you how the binding energy is calculated from Autodoc. If you see the binding energy, okay, we generally calculate the change in Gibbs energy, whatever we studied in thermodynamics. We know that the change in Gibbs energy is the change in enthalpy to the difference of the product of temperature and change in entropy. If this is the formula for change in Gibbs energy, this is another formula with respect to standard Gibbs energy. That is, change in Gibbs energy is the change in standard Gibbs energy at standard state. What do you mean by at standard state? So this is before the reaction. This is after the reaction. So whatever I have given is before the reaction. So after the reaction, you can able to see it's going to have a rate constant. You remember yesterday I was mentioning you about the enzyme substrate. If you react a substrate with an enzyme, we'll be getting an enzyme substrate. Okay. If you take a general concept like ligand receptor, okay, we are going to find the ligand receptor. So in this case, whatever we associate the ligand and receptor, the corresponding rate constant what we calculate is association constant. Generally, the association constant is the product of output to the input. What is the output? Ligand receptor is the output. And so the input is the product of ligand and receptor. If this is association constant, okay, under reversible conditions, can be a pH change, can lead to form dissociate the ligand receptor into ligand and receptor. Under that case, just because it's dissociating into two molecules, this is Kd. So we measure the rate constant as Kd, and the Kd is again the output to the input. The output is nothing but ligand receptor, and the input is the ligand receptor complex. So this is how K and Kd can be measured from the reactions. So this is before the reaction, whatever given energy we calculate. R is universal gas constant and T is the absolute temperature. Kd is dissociation constant. Got it? Now, let me take you to the molecular docking. So, molecular docking we have already seen, just docking a ligand and receptor. But what exactly is molecular recognition in molecular biology and how we can link with this molecular docking? Molecular docking is not merely a docking of a small molecule ligand with a receptor, but we also find a macro, like a DNA and RNA can also bind with protein. Still, we call it as molecular docking. Then we can see a few examples of biology. For example, if you see antigen binding with antibody, in which subject you can see this antigen binding with an antibody in immunology, isn't it? In immunology, this is most important because this antigen antibody affinity is going to give more than 99.9% accuracy on treatment, especially if the patients are advancing in the late stage of cancer. Okay, instead of treating with cellular immunity, the clinicians prefer to treat with humoral immunity. Humoral immunity, it means that treating with antibodies for cancer, at least it can increase a few days lifespan. Okay, the antibody binding with the antigen, the antibody generally binds with the antibody. There is a region called paratope. The paratope region of the antibody is generally going to interact with the epitope region of the antigen. And this kind of a binding affinity, whatever we study in immunology, is of course a kind of a molecular docking. Whatever we call this molecular recognition is a talking. And now let me come across the enzyme substrate. We already seen plenty of reactions and I illustrated you. So this comes under enzymology, the branch of study where we say study about the different enzymes or different substrates binding the enzyme comes under enzymology. Now transport molecule. Can someone give me an example for this? What example we can take? Here we can take TMB substrate and plenty of enzymes binding with the substrate. How about this? Transport and molecule.
Sir, lactose binding to the permease in E.
Exactly.
coli in cobra.
Good one. Very good. Good example from your side. I can give a simplest example like oxygen transporting through hemoglobin. You know that oxygen, if it during the inhalation, it's going to mix with the hemoglobin to form oxyhemoglobin. Oxyhemoglobin is of course a different protein. Hemoglobin is different. Oxyhemoglobin is different. Similarly, during the exhalation, that is, we see that CO2 is releasing from the hemoglobin. So during that time, carboxyhemoglobin. So in this case, the carrier molecule, what I mentioned here, is the gaseous molecule, either oxygen or carbon dioxide, through the transport protein called hemoglobin. Okay, so this also comes under signal transduction. There is a subject called signal transduction where we discuss more about these kind of different types of signaling. And here in this example, signaling ligand binding with the signaling receptor. The best example I can provide is the acetyl choline. How our brain works actively. So if an acetylcholine chemical is going to bind with a neural receptor, obviously can find a neural signal. So that's what we can see the brains, billions of neurons gets activated and we can see full functioning of brain. Okay, so this is something about different examples of biology which are linked with the molecular talking. Now related to understanding the molecular recognition. So whatever we discussed, the relevance of molecular biology with molecular computational molecular talking. Similarly, we can link the computational talking with pharmaceutical terms. For example, here the drugs can be agonist. Okay, generally whatever we use the drug for treatment of diseases, that we call it as agonist in pharmaceutical technology. If that is agonist, there is also one more term called antagonist. The antagonist is going to give the same drugs can give adverse effects. Instead of giving a desired biological effect, it may give undesired effects. So I can tell you some examples like if you are using a neem leaves, you know that neem leaves are best example for treatment of cold. So if you are using the neem leaves, you can able to see around 500 compounds can be present, but not all the 500 compounds can give a positive effects on treatment of cold or cough. Few compounds can give even side effects or give some undesired biological effect. So that is what we call it as antagonist. Okay, and generally, if you see inhibitors, inhibitors can are of different types based on types of inhibition, competitive inhibition, uncompetitive inhibition. Okay, and we have mixed inhibition. There are types of inhibition, isn't it? So based on the types of inhibition of a ligand with a receptor, we can have the types of inhibitors. So this is something about different technicals or technical terms or terminologies we use in talking. But you know one thing, we don't know if you have a ligand and if you have a receptor. So if you can just determine the binding affinity alone through binding energy, we don't know whether it works like an agonist or antagonist. So molecular docking merely tells you the binding energy. It won't distinguish whether it can give an agonist function or antagonist function. So then how come we can determine unless we work in vitro studies or in vivo studies, we can't confirm the same. Now we discuss on theory of molecular docking. So if you see the theory of molecular docking, the first theory was proposed by Fisher on lock and key in 1890, and Daniel Koshland proposed the second theory called induced fit. Nowadays, whatever tools we are using, starting from freeware, Autodoc to commercial tools like Discovery Studio, Scroll all these tools uses conformational ensemble model for docking. Now about lock and key. So if you have a key and if you have a lock, see the key which is binding with the lock region. This is not going to undergo any conformational change. It's not going to undergo any conformational change, just like that we are fitting. Okay, that's what lock and key model. The induced fit model, if you have this as a ligand and this as a receptor, so during the binding, both the ligand and receptor conformations are changed. So area, the shape is different for receptor and similarly for ligand. Now the shape is entirely changed based on binding. This is what we call it as induced fit theory, which was proposed by Daniel Koshland. So in this case, what we understand, both the ligand and receptor are in flexible state. If and if it is flexible, then only during the docking, both the ligand and receptor can mutually fit with each other during induced fit theory. Now, let me take you to the conformation ensemble. In conformation ensemble, this is the third model. In this model, we can see that a ligand is going to bind with the receptors. If ligand is binding with the receptor, if ligand is binding with most appropriate binding, that is the best fit, then you can find negative maximum energy. So out of different conformations, for example, a ligand one, if it is binding here, if it is giving an energy like this, but if it is docking somewhere here, if it gives a most negative maximum energy when compared to 1, 2, 3, 4, 5, and a number of binding conformations, then I can tell that this second conformer is showing the local energy. Whatever energy we get, the local energy is only one. But whatever, sorry, am I telling something wrong? Yes, this is what we call it as global energy. So the two, whatever we find is the global energy. And whatever we find in conformer 1, 3, 4, these are, we call it as local energy. Global energy will be only one. The local energy can be multiple. Generally, we check for the best global energy on binding with ligand and receptor. So this kind of a method of determining the energy in different sites of binding is what we call it as conformational ensemble method. So if you see to the lock and key to the ensemble model, we are using presently the conformational ensemble method. And in docking, you can see the types of docking. Like as I mentioned, small molecule can also be docked. Small molecule protein. Similarly, protein nucleic acid, protein, small molecule nucleic acid. All these things comes under docking. So what exactly we can able to find in docking? So the docking of ligand and receptor is not merely fitting like a puzzle. This is not merely shape complementarity. Most of the tools, okay, are using the chemical complementarity. What do you mean by chemical complementarity? Then how this piece is going to fit this piece? For this, we can see examples here. For example, if your ligand is going to have hydrogen bond acceptor, and if your receptor is having an hydrogen bond donor, okay, so that is hydrogen bond acceptor, I mean OH group. I told you the red color thing, whatever we find is the OH group. The OH group can bind with the H group. So you can able to see this kind of a binding forms a hydrogen bond interaction. So whatever we can see, this dotted line is a hydrogen bond interaction. So this is an aliphatic group linking with the aliphatic receptor. I mean, aliphatic hydrogen bond acceptor is sharing a single hydrogen bond with the aliphatic hydrogen bond. In this case, you can see the aromatic ligand is binding with the aromatic amino acid of a protein. You know that we have aromatic amino acid residues even in amino acid residues. So suppose if your ligand is binding with the aromatic amino acid, it can able to share 5 to 6 hydrogen bonds based on whether it's a 5-membered ring or whether it's a 6-membered ring. Okay, so based on this, you can able to see. So if you have 5 or 6 hydrogen bond interactions, definitely the aromatic substitution, the aromatic kind of interaction is best when compared to aliphatic. Because we have, if it is 6, we have 6 hydrogen bonds, which is more stable when compared to the aliphatic interaction. Similarly, if you can also able to see a slight difference with respect to electrostatic. What do you mean by electrostatic? If positive group of ligand is going to interact with the negative group of amino acid of the receptor, then we can find an electrostatic interaction. This is somewhat stable when compared to aliphatic interaction, but this is unstable when compared to aromatic interactions. The next thing is the hydrophobic. The hydrophobic, it means that lipophilic interaction. What do you mean by lipophilic interaction? So if you can able to see the lipid molecule is interacting with the lipid molecule, it can able to give this hydrophobic kind of an interaction. This is what we call it as chemical complementarity. Now, let me take you to the energy. Like if you see the energies, as I mentioned earlier, we have a receptor binding with the ligand. The ligand is shown in the green, and the receptor is shown in the blue color. So if you see this receptor ligand, so this is the first conformer, this is the second conformer, and this is the third conformer. See the shapes how a ligand is binding with the receptor. In this condition, the ligand is perfectly binding with the receptor, isn't it? When compared to the other states. So whatever energy we get in the third, that's what we call it as global energy. The remaining energy is what we call it as local energy. Now let me take you to the further thing. Molecular flexibility in protein ligand docking. If you see docking, ligand protein docking, the only one docking which is not favorable is flexible protein and rigid ligand. This alone is not possible. If this is not possible, what are the other possibilities you can find with respect to rigidity and flexibility of ligand and receptor? So rigid ligand, rigid protein can bind with each other. What is this we call? We call this as lock and key model. You remember? But at present softwares, what we exactly use? Flexible ligand and flexible receptor. So this is more complex, but most of the freewares and commercial tools are developed like this. Got it? And this you can able to see in induced fit theory, and also you can able to see it in conformational XML methods. Now let me take you to the docking. So if you take you to the docking, now you can see an animation. The ligand is docking in different poses. So this might be the first base pose, but we can see other poses. So the ligand is undergoing a conformation binding with different sides here, somewhere. Okay, the earlier it was not like that, but now it's going to bind. So this is what we call it as conformations. Conformation one, conformation two. Okay, can have different conformations. Okay, and now if you see this kind of a talking I told you earlier, not only we find the aromatic kind of interaction is best, but also the hydrophobic. See the hydrophobic how it goes. Hydrophobic, I told you it is a buried site inside the protein. So whatever sites you find on the surface of protein is hydrophilic context. But if any uh, protein is buried inside, okay, if a region is buried inside, then the contacting of ligand inside the buried site is very difficult. But in some cases, we have to target the buried sites. So whatever ligands we design is of hydrophobic in nature. That is, we design the lipophilic ligands. Okay, and you see the electrostatic. This can give a good interaction when compared to the hydrogen bonds. This interaction is more stable because of ion, the negative ion and positive ion is going to interact, and you can find the electrostatic interactions in this case. Now, how the binding sites are determined. So you see that a protein molecule is packed inside a box, grid box, and now we are filling it with water. Yesterday I mentioned, isn't it? And wherever the water is going to accumulate on the surface, hydrophilic binding sites are detected. If water is not getting inside the protein, but you have a shallow hole, then it is a hydrophobic binding site. And this is what we are using in Discovery Studio Ligand Fit, flat fill algorithm, filling with water and finding the binding sites. Now let me take you to the scoring methods. If you see the scoring methods, okay, we can able to see the scores or generated from the interaction energies between the two molecules. What are the two molecules? Ligand and receptor. So this is also can be determined through the desolvation and solvation energies. This is another parameter how the scores are calculated. The third is the entropic factors. Entropic factors that occur upon binding. Entropy, sudden change in energy, sudden release in energy. This is what called entropy, isn't it? The entropy factors can also decide the binding affinity. So these are the journal papers I've shared you earlier. That is working on computational study on that is how I worked in this study. This is not exactly work by me, but this are work by professors in Rajalakshmi Engineering College. They secured a grant from UGC. Okay, and then they worked, and myself I worked on computational part and even the publication part. If you see what exactly we have done, we have taken around 8 different target receptors for cardiovascular diseases. Then, for the 8 receptors, we targeted with a plant compound called vitaferin, which is a steroidal lactone from ashwagandha. And for these 8 receptors, we have 8 different control ligands. Okay, and after talking, we can able to see epinephrine A is showing the better result with these two receptors. Epinephrine A, the plant compound, is better talking with these two receptors out of eight receptors of cardiovascular. Then we have finalized that this compound can also be used for treatment of cardiovascular diseases by binding in these targets. Okay, so this is a kind of a study where we have used molecular talking and the simulation. So these are the two things we have tried, MD simulation. And this 3D interaction of ethafenate talk with the BRP1 receptor of heart and P2Y1 cells. The next study is this was simple studies and it's easier to understand. So what I'm first explaining you with the easiest uh, publications. So molecular talking and simulation binding analysis of pyrimidine. The pyrimidine is coming under protenoid. Protenoids are also called as isoflavones. Generally, these are used as a pesticide. But if it used in optimum level, this can also be used as a drug for treatment of liver cancer, especially the primary liver cancer, like hepatocellular carcinoma. So in this, I tried with five different receptors. Out of five different receptors of HCC, the this plant compound is talking well with these two receptors. So I finalized and we have shown these results. Okay, so talking through Autodoc and even the previous study we have done talking through Autodoc. So you can able to see whatever we have talked with Autodoc, we can able to publish in at least Q3 journals. Even now we have Q1 journals published with whatever we published. These are we have tried with Autodoc. You know the difference between the pictures whatever I shown you earlier. The earlier whatever I shown talking of again with receptor is a 2D diagram. I mean 3D diagram. But this is a 2D diagram. You can see this is the ligand here, and this ligand is interacting with these amino acids. Okay, exact amino acid if you see glutamic acid 257, it is sharing the hydrogen bond. Hydrogen bonds are shown in green color. And whatever you can see, this 2.6 is the hydrogen bond length. So like this, if you see the glutamic 257 is interacting with two hydrogen bonds, the ligand. Okay, there is another amino acid called lysine 293, which is sharing one more hydrogen bond. Okay, so this is something about the talking images. We can get either 2D or 3D. And the 2D or 3D we can get from even Discovery Studio Visualizer. The freeware is sufficient enough to provide this kind of. Now let me take you to the talking flowchart using Autodoc. So what are the steps in Autodoc? If you see in Autodoc, okay, the majorly we should prepare two types of files. One is ligand file and another is protein file. So the ligand file and protein file both we have to convert initially into PDB-QT formats. Like however we have tried with Autodoc PYRX, here also we have to convert like that. After converting these files, then only we try to convert a TPF. Initially, we convert GPF file. GPF stands for grid parameter file. So we can create this during the grid generation. After running the grid, we call GPF, and after running a tool called talk2, we'll be getting like TPF, that is talking parameter file. We also have grid log file, so we find so many intermediate files are developed during the talk. So more about this if you do practically you can understand well. Now let me take you to the protocol. This is the Autodoc protocol. So I suggest everyone to practice initially with the demo files, whatever I provided. I mean whatever I've shared. Now let me take you to the files. Okay. So actually if you see this, I can also tell you like how many steps are there on these things. Okay, let me come outside because I have less time. I have to show. Let me take you first to the folders. So whatever folders I have shared you, so Autodoc installation files. If you click here, so you can able to find these files. Autodoc suite. So one, if you want to practice with Autodoc, you must install all the four tools: Autodoc Suite, Chimera, EDU Pymon, MGL tools. So these four tools can be installed in random, but all the four tools have to be installed. So whatever I have provided are compatible with 64-bit version. Not only we are going to install these 5 exe, I mean 4 exe files, but also download this file. This is actually an Autodoc tutorial. So if you want to perform the steps, you can also download this tutorial along with this installation files. Okay, of course it will be needed for you. So after that you can see Autodoc demo. In Autodoc demo, we should create one folder, okay, in a desktop, and we can we can download the entire thing like tutorial, Autodoc 4.exe, AutoConnect 4.exe, and this is a sample LeCan file. This is a sample routine file. So all these things we can download in a single folder for practicing. Okay. Now I'm going to download and I'm going to show you how I'm going to do it. So I told you for installation, use this Autodoc installation folder. For practicing, use Autodoc demo. And Discovery Studio Visualizer is the older version, whatever I uploaded. But the latest version takes much memory. If your CPU is of less memory, you can install this Discovery Studio. Or else you can download and install from BioVia. Okay, so you can find the latest features there. Now let me take you to the illustration of Autodoc. After installation of 4 exe files, what are the exe? I mean icons I find, this is the icon, most important icon. Uh, so second icon present in desktop is this, and the third icon, this is Python molecular viewer, Autodoc tools from MGL, computer-aided track design. This icon, and fourth icon, we get like a green color thing. Yeah, the vision. Okay, so these are the four icons you get after installation of 4 exe files. For Autodoc PYRX, whatever I showed you earlier, we get these icons. But for Autodoc, we have to get all these four icons. If you want to start with Autodoc, simply double click. Now the Autodoc is going to be open. Before that, we can create one folder. So if you see this Autodoc, whatever I'm going to show you a demo, this is of course a commercial tool. But initially, if you are installing, then for 15 days it works. It can work for free. Okay, it won't. You won't get this kind of window. And after 15 days, it asks you to register now. Instead of clicking this, you can use remaining data. Even if you click this, you can able to access the talking tool. So this is the window of Autodoc. This is the window of Autodoc. So here we are going to use this for talking purpose. So for initially docking, we have to create a demo folder. So I create a folder called demo 10. So in this demo 10 folder, I have to download all the files, whatever present in Autodoc demo. So first try with the demo files. Later you can try with your project files. First I'm going to download Autodoc tutorial. Okay, so where I'm going to save, in desktop demo 10. Then I'm going to save Autodoc 4.exe. Download. It asks for dangerous. It mentioned it. So no, I'm practicing. There won't be any problem. Just like the download, because the exe files are obviously, or most of the exe files or virus files. In Linux, you can't able to download just like that. Only in Windows it's possible. Okay, we have Intinavir is the anti-HIV drug, and Protease is the HIV protease. So today I'll be providing you a demo on how whether this Intinavir is binding with the HIV protease. So in Autodoc, okay, I'm going to illustrate you Autodoc 4.0. Here, if you see this, we have steps like the first step is prepare the protein file. Second is prepare the ligand. Third is prepare the flexible residue. In each and every step, main steps, we have substeps present. The fourth is running autocrate. Fifth is preparing the docking parameter file. The sixth is reading the docking log file and analyze the results. So we do up to this. The seventh step I'll be teaching on tomorrow. Today we'll be practicing on the first six steps. Okay. I start with the step number one. Before starting with step one number one, I'm audible to everyone. Yes or no?
Yes,
Yes,
sir.
sir.
Yeah, let me go ahead with the first step. So for the first step, you go to the window, go to the file, preferences, set. Inside the set, so wherever we have that folder, we have to give it in startup directory. We already have a demo one folder in the desktop, but we have a demo team. So I going to paste the exact URL where I kept all the input files. That is demo thing. I have typed it now in startup directory. Next step is click make default and then next click set. So this is the first step we have to make to set whatever intermediate files we generate during logging, it will be stored in that specific folder. For this, we are taking the first preliminary step. So after setting the directory in the startup directory, dismiss. So now we start from here. After setting this only, we are going to. The next step is load. First step is prepare the protein file. For this, load the protein file. Read molecule protease open. Now the protein molecule is open. The first step is completed. Okay. Next, edit hydrogen. Edit all hydrogens. Edit hydrogens. Add default it should be all hydrogens. Click OK. So after adding hydrogens, you can see that white color bonds are appearing on the surface. The next thing is edit add Coleman charges. Edit charges and Coleman. So we got Coleman charges. Simply click OK. So up to this step we have completed. The next is file save write PDB. File save write PDB. So during this step, you can save this file as protein1.pdb. So just whatever I mentioned, you follow exactly. If you are not following any one intermediate step, you won't get the result. So follow exactly like however I am providing in the demo. Later you can try with your sample files. Click OK. Now I have completed up to this step. The entire first step is completed. That is, I have prepared the protein file. The second is prepare the ligand. For this, ligand input open ligand. Ligand input open ligand. In the open ligand, I have to open the ligand, but the ligand is not available. The reason behind is here we got like PDB-QT file, but the ligand what I mentioned is in PDB format. So I select all files. Then only the ligand is visible now. The ligand is engineer. Simply click open. Now you get this window, simply click OK. Okay, the ligand is loaded. It doesn't mean that ligand is talking with the receptor. The ligand is just loaded in the window. Okay, you have to take steps in talking later. So I have completed this step alone. The next step is ligand torsion tree detect root. Ligand torsion tree detect root. The step is completed. The next step is ligand output save as PDB-QT. So now only we are saving ligand output as PDB-QT. Initially it was ligand Indinavir. Now I'm going to save like ligand1.pdb-QT. This is how I'm going to save the ligand file. Now this second step is completed. Only the ligand alone we have prepared as PDB-QT. The third step is to prepare the flexible residue file. For this, go with grid macromolecule open. Grid macromolecule open. So if you click open, we have to open the macromolecule. But we find only the ligand. Then how to open the macromolecule? See the PDB-QT file so that we did convert protein as PDB-QT. Unless we convert, it won't open. So what I do is click all files. See the latest protein file what I saved. The latest protein file I have saved is protein 1. Select the protein 1, click open. So it prompts with a window, simply click OK. Now see, it's already automatically getting a window to save a file. Now only I am going to save this file as protein1.pdb Here in the second step, we have saved ligand1.pdb. In third step, this is the first time I'm going to store I have saved the protein in the form of pdb format. Click save. So after this step is completed up to this step. But we have even a part of a step in part 3. That is defining active sites. How to make it? Select from string. Select, select from string. So here we have molecule list. Click this molecule list. Select the recent protein molecule what we have saved. The recent protein molecule is protein 1. Select that. And this protein is having a single chain. If you're talking with the ligand with the receptor, I suggest you take a single chain. But you don't know where exactly the binding site is present. So you simply have to type asterisk. Asterisk, it means that the binding site can be any of these atoms, okay, in a protein. So single asterisk is sufficient. Remember, we have to click only once add. If you click two times, or if you forgot to click one and you again two times unknowingly click, then this docking won't happen. You have to close and you have to once again download the files whatever I provided in the drive, and then you have to try. Okay, got it. So now just because I've done all, I already clicked that, click this once. So this part, the entire third part is completed. Now go to the fourth step, running autogrid calculation. Grid set map types. So grid set map types. So wherever you find choose again, instead of choose again, I suggest you to click open again. So grid set map types open again. Only we have to open again. So again one.pdb. So I have completed this step. Okay, instead of choose alone, I have used open again. Okay, this step is completed now. The next step is grid box. Grid box. See this box, a small box is open inside, but I have to enclose the entire protein in the box. So I'm going to increase the dimensions. I'm going to increase the dimensions, but you can able to see the values for the maximum you can increase up to 126. So click apply. 126 is there. I'm going to give a value like 120 for the red color dimensions. So now just stretch to 120. You know what is 120 here? See here, this red color alone stretch to 120. Now I'm going to touch the green color. So this green end should be enclosed. The green is smaller region, isn't it? So first click apply. Now I give lesser thing like 100. So even the 100 is covering the entire region. No problem. Okay. So if you take much of the region, then it docks very slow. I like to dock as soon as possible. Whether any molecule is coming out, no. What if we have to check with the blue color? Now apply and OK. This is okay. Now blue color. The blue color we can slightly increase to 126. First click apply. Everything is getting activated. I give like 126 apply. Yes. 126 is still maximum. I reduce something like 120. Yeah. You know, we have packed the entire protein molecule inside the grid box. If I rotate in all the directions, still don't find. Okay, this blue is not enclosed. See, then you have to increase the blue. Not 120 is not sufficient. I increase even the maximum as 140 and value also as 140. Now I enclosed everything. Okay, click OK. Now you can rotate in all the direction. So the ligand is the receptor is perfectly packed inside the cubical box. So after completing this step, file, close, saving. Correct. That's it. So I have completed this step. Now the next step is, up to this we have completed. The next step is create output save file as GPF. Create output save GPF. Now you are going to save this file as grid1.gpf. GPF, I mean grid parameter file. I'm going to create a grid1 as a grid parameter file. So after forming this is the step. Run autocade launch and run autocade. So you have to see that autocade 4 is there, but the entire URL is not there. So click browse. If I click browse, default it take you to the demo 10 folder. Auto grid has to be selected. Auto grid 4 exe. Click open. See, now you got the entire URL. Now similarly, in the parameter file we have grid1.gpf, but we don't have URL for now. So click that grid1.gpf browse. Grid1.gpf select. You can also see the grid block file is automatically created now. Simply you have to click launch. Now running the autocade is happening. So if the white window immediately closes, then there is some mistake you have done in intermediate steps. If white window is not closing immediately and it's taking some time to close, then your whatever steps you have taken is correct. This is how you are sure confirm whether your talking protocol you have carried is correct or not. Yes. After some time only it closes. You are fortunate to move to the next step. The next step is preparing the talking parameter file. That is talking macro molecule set rigid file. Talking macro molecule set rigid file name. So macro molecule. So we have to select only macro molecule protein one. Open. So after completing this step, talking ligand open. Talking ligand open. So only the ligand file has to be open. So click open. Now we are getting this window. T for lead. We have to accept this. That's it. We have completed this. Next, talking search parameter genetic algorithm. Talking search parameter genetic algorithm. So you have like runs. Okay, 10 runs. 10 runs, it means 10 conforms. A ligand is going to bind in 10 different sites. But there is no need. If a conformer can be able to bind in a one, two based conformers, that is sufficient. Then I can reduce the number of genetic algorithm runs as 2. Click accept now. So to reduce the time of talking, I reduce the GA runs to 2. If you take 10 runs, it takes 5 times the time. Now I have completed this step. Talking output American. Talking output American genetic algorithm. So now only we are going to save a talking parameter file as doc1.tpf. So we are saving a file, a talking parameter file now. So this step is complete. The next step is run Autodoc launch. We need only 2 minutes to complete. Run Autodoc. Here in Autodoc 4 we don't have URL, so click browse. Select the Autodoc 4 whatever we have downloaded. You get an URL. Doc1.tpf don't have URL. Simply click this. Select this. Select this. Enter file location up here. After finding the file location in both the DPF and the talking log file, simply click launch. Now we are getting a white window. So if that white window closes, the final step is to so it didn't close. Actually, just use the tab. We have to use the 6th step. If only if you complete the 6th step, then if you even if you close the window, tomorrow I can open the window once again and we can do the analysis. If you close the window in between, then again you have to start doing from the beginning. So let me complete even the 6th step if the window closes. In the 6th step, what I'm going to do here is the ligand is already docked with the receptor in this step. So this ligand receptor I'm going to save as PDB file, ligand receptor complex.pdb. So that I am going to do in sixth step. Fifth, the fourth step we have run the autoclave to determine the binding site. The fifth step we are running the autodock to dock a ligand with a receptor. So the final step is to save the ligand receptor. Right. So this is what we are going to do. I have chosen smallest ligand file and smallest protein file. Now the window is closed. So we are getting results instantly just because the files are very smaller in size. So for demo purpose, we can able to use these files. Now let me take you to the next step. Analyze dockings open. Analyze dockings open. So in analyze dockings open, we have to open the doc1.dlg. So you'll be getting this window default. Click OK. Click OK once again. Next, analyze macromolecule open. Analyze macromolecule open. That's it. The step is completed. Now let me go to the next step. Analyze conformations load. Analyze conformations load. See, this is most important step. Here we got two conformations. The first conformation is minus 6.28. So if the energy is minus 6 and below, the docking is significant. So in our case, whatever we talked in International protease, the first conformer alone is significant. So not only we got the binding energy, we also got inhibitory constant value. That is what is the dosage of Indianaware one can able to choose. The dosage they have mentioned is 24.84 micromolar. So this is inhibitory constant value. So if you want to save this, you can save this in a paint or something like that. Okay. So later we can input this results. Of course, this is also present in interpreted files. But if you don't want to forget this, okay, you can save this in that as a JPEG format in the desktop, the demo 10 folder. So I just saved like doc scope or binding scope, whatever it is. Okay. The next step is analyze conformations, play rank by energy. Analyze conformations, play rank by energy. We get like an ampersand sign. Simply click this ampersand. We'll be getting one more. Click build current. Click right complex. In the right complex, you get like PDBQT file. We have to select as more because we want only .pdb file. The ligand I used is like Internaware, so I'm going to use like Internaware. Protease is a protein receptor, so I'm going to give like protease underscore complex. Internaware underscore protease underscore complex dot pdb. So this I'm enclosing it in double quotes because I want to save it as dot pdb file. So this is a ligand receptor complex in pdb file format. If you have saved up to this, now you can close out of it. We have talked and we have saved that file as .pdb. Tomorrow we'll be discussing on how to analyze the results with that saved pdb file. So instead of providing demo on Bimol, I'll be telling you how to analyze the results using Discovery Studio Visualizer because that is very easier to analyze the results. So more about the docking part, analyzing results, we'll be discussing on tomorrow. And also tomorrow we'll be discussing on how to perform MD simulation and what are the different analyses we do in MD simulation. So today I have completed the session on molecular docking and I have also provided you on the fundamentals like how to pursue docking using Autodock. Any doubts are welcome to ask.
Sir,
Yeah, tell me.
sir, I tried installing Discovery Studio, sir. I was facing some difficulties. I even shared the screenshot with you to your WhatsApp
Yeah,
number,
but
sir.
how come you have to share the screen now? So I can't able to clear the doubts unless you come in the online and you share your window. Okay, I can't able
Oh,
to clear your doubts. So either you can make it now or on tomorrow if you join on the at time at 7 o'clock. If you are ready with your laptop, you share your screen. Okay, I close my screen. Then I can illustrate you how to make it. Then I can start with the docking analysis. However, it's feasible. You can try it. When can we
Okay, sir.
try?
Then I'll come to tomorrow with a screen online, sir. Right now I'm
Uh,
in
uh,
travel.
yeah. But at 7 o'clock, you have to log in sharply. Okay.
Yes, sir, sure, sure. It was asking for some password. That is why I called you.
There won't be any problem. I can tell
Okay,
you
sir.
how to skip from the password. So
Okay, sir.
tomorrow I clarify it. May I know your
Okay,
good
sir.
name?
Thank you so much. Sir, I'm Angela. Angela Joseph.
Okay, your name is not audible. Okay, no problem. Tomorrow will be coming, isn't it? You can say. Thank
Yes,
you.
sir. Okay, sir. Thank you so much, sir.
Any other doubts participants have, you can ask. Like not only this issue, even if you have any issues and you want to share the screen, you are welcome to ask and to clarify all these doubts at any time. So be ready with your laptops. Okay, I can able to clarify.
Sir, one doubt,
Yeah,
sir.
tell me.
Sir, molecule binds at the lowest energy, right, sir?
See, your voice is feeble, often breaking. Again, tell me. Ask me once again.
In molecular talking, the molecule binds to the receptor at the lowest energy possible.
You are right. You are right.
Uh, it means that energy is absorbed or energy is liberated.
Actually, see, we are just checking whether the ligand is the drug is going to bind with the protein or not. If the drug is binding with the protein, we can able to see we call the drug as a bounded drug. If drug is not binding with the protein, then we call it as unbounded drug. You know, I explained you something about active pharmaceutical ingredient in the drug. 10% of the API is the active part of the drug, which is going to give a biological effect, biological response. If so, if not all the 10% of the drug is going to bind with the desired site, some can also bind with antidesired sites. So under that case, even the drug can also unbind. And the excess of the drug, which is not bound with the receptor, the exactive binding sites, that will be washed away, that will be excreted away through the kidney. Okay, so that's what we call it as unbound drug. Okay, so now you ask something like binding energy. In binding energy, you should have to understand that if a ligand is binding with the receptor, okay, we understood through the energy should be negative maximum. For example, if you are speaking, you are releasing the energy. If I am thinking and answering you, I am burning somewhat extra energy when compared to you. Okay, so even sleeping, we are releasing energy. So whatever we do, the catabolic reactions, you are doing some actions, you are doing some kind of thinking. All these things release energy, and in all these cases, the energy is negative. So if negative maximum means you are releasing maximum energy. So if a drug is binding with the receptor, then we have to get negative maximum binding energy. So if you are getting somewhat maximum energy, if you, in my case, I got minus 6 point something as a conformer one energy, and in conformer two as minus 4. The minus 4 is not negative maximum, and the two it is more when compared to minus 6 kilocalorie per mole. So it is not significant. Got it? So we humans release energy through catabolic reactions. Similarly, even drug binding reactions or catabolic in nature. Anabolic is generally like photosynthesis reactions. Okay, the drug binding is a kind of a catabolic reactions. So it has to dissociate. Okay, anything else you like to ask?
So it is basically like some energy is spent to the molecule for it to bind with the receptor. After binding, the energy will get released.
Yeah.
Ah, understood.
Okay, thank you. So any other doubts you can accumulate and ask me on tomorrow. Okay, the parts. Now let me finish this session. Good night to everyone. Good interaction. Thank you very much. Bye.
Thank you, sir.